Assalamu alaikum. Welcome back to respected participants. Now it's time for the second lecture of today's session, and our respected speaker, Dr. Wasim, is here. I request Dr. Wasim, please, you can share your screen with our respected participants. And now, formally, I invite the Gen Ambassador, Ms. Asma Talib, to introduce our respected speaker, Dr. Wasim Akram from HBRI NARC Islamabad. Thank you.
Thank you, Dr. Saqib. It's my pleasure to introduce our distinguished speaker, Dr. Wasim Akram, Senior Scientific Officer at Honeybee Research Institute, NARC Islamabad. Dr. Wasim holds a PhD in entomology from the Islamia University of Bawalpur, with the research focuses on native bee diversity, pollination, and honeybee health. With over 17 peer reviewed publications and 6 book chapters, his scientific work addresses critical challenges in apiculture from integrated pest management of Varroa destructor to the toxicity of insecticides on honeybees. Dr. Wasim has involved in ALP funded research projects and currently supervises PhD and internship students in apicultural sciences. Dr. Wasim brings extensive hands on training experience. He has organized and served as a resource person for 12 plus national training programs on beekeeping, including beginner courses, honey processing, export quality honey sourcing, and IPM of honeybee colonies at NARC. He also coordinated a beekeeper convention for expert networking with Dubai, UAE, and recently completed advanced international trainings in drones, IoT, and application in agriculture at Hosang Agriculture University, China. Today, Dr. Wasim Akram will deliver an insightful session on honeybee health management, entrepreneurship, scientific and commercial strategies for disease and pest control services, highlighting the scientific principles and entrepreneurial opportunities associated with honeybee health management. Dr. Wasim Akram, we warmly welcome you and invite you to take the floor.
Thanks, madam. First of all, I am very thankful for this opportunity to speak in this webinar today. Honeybees are the most important pollinators throughout the world because they produce honey. They do pollination. They provide pollination services and conserve our biodiversity and conserve our bio. But however, a lot of pests attack honeybees, which ultimately cause economic losses to beekeepers. In this presentation, we will discuss the major pest of honeybees, their damage symptoms, and their management practices. Here is the list of pests. The first one, wax moth. There are two types of wax moth. One is the greater wax moth and second lesser wax moth that are distributed throughout the beekeeping areas in Pakistan. The second pest of honeybees are mites. Two types of mites. One, ectoparasitic mites. Ectoparasitic mites are those mites that feed outside the body of the honeybee, which are Varroa destructor. And second ectoparasitic mite is Tropalileps scleri. And the other type of mites are endoparasitic mites like Acarapis woody, which is called tracheal mite, that live inside the body or tracheal system of the honeybees. Next pests are hornets. There are 5 types of hornets that prey on honeybees. Uh, some minor pests like ants, birds, hive beetles, black bears, lizards, frogs also attack on honeybees. In this presentation, we will discuss the major pests one by one. The first one, wax moth. The distinguishing character for the identification between lesser wax moth and greater wax moth is the yellow head. The lesser, you can see in the picture of lesser wax moth. Lesser wax moths have yellow head in the front, whereas the greater wax moth don't have the yellow head. The greater wax moth are brownish gray in color, with heavy bodied, whereas the lesser wax moth are silver gray in color and not heavy bodied. These species of wax moths cause are serious problem in the hot and humid weather condition. They attack during the monsoon season, but remain active from March to attack on old cones, which are black in color and weak colonies. The female wax moth enter through the cracks and crevices or through the main entrance of the hive and lay eggs on the bottom board. You know, when a bee emerge from the cell, she chew a worker bee or drone bee when emerge from the cell, she chew the capping and drop down on the bottom board. The nurse bees also perform the cleaning duties. They during the cleaning of cell, they remove the pupal skin, molted skin, and drop down onto the bottom board. These uh, these material these material provide the breeding source to the wax moth on bottom board. When the egg when the egg hatches, the larva emerge and start feeding on the material that are fallen on the bottom board of the hive. And when the larva grows, climb to the shifted towards the frames or combs and start feeding. Either the frames are um, food frames, it means pollen or honey frames, or drone brood sheets or worker brood sheets. They make silken tunnels, and as you can see in the picture, how the larva feed and make silken tunnels. Um if if the larva attack on cap brood, they chew the capping of the cells and block the cell with the silk, due to which the development of the bees the cell. In case of serious infestation, they cause complete destruction of the colony, or sometime the bees abscond. Here in this picture, you can see how the wax moth attack on pollen frame. Here's the mild damage, moderate damage, severe damage, and complete damage. As I have already mentioned, they mostly prefer to attack on older combs. Here in this picture, you can see how they damage the older combs, and you can see the foundation sheet is not affected by the larvae of wax moths. Here's the damage pattern of lesser wax moth, almost similar to greater wax moth. Here's the management. Two types of management practices. One is the preventive, and second, curative measures. In preventive measures, keep the colony strong, supply adequate quantity of food, either in the form of pollen patty or sugar solution. In case of severe infestation, reduce the hive entrance and seal all the cracks and crevices. The most important, clean the bottom boards of the hive every 3 to 5 days. Otherwise, the bottom board will provide the breeding source to the wax moth. And remove extra frames and store properly, as you can see in the picture. Then curative measures. You can kill all the stages of the wax moth larva by placing the infected combs at -6.7 degrees Celsius for 4 to 5 hours, -12 degrees Celsius for 3 hours, and 15 degrees Celsius for 2 hours. As well as you can also use microbial pesticides and biological control agents for the control of these pests. Uh, uh, the Bacillus thuringiensis based pesticides and fungus based pesticides such as Metarhizium anisopre and Beauveria bassiana And you can also use biological control agents like Trichogramma species and Apetanlis galeriae. But you have to apply these microbial pesticides and biological control agents only in those bodies or those areas where you have stored the frames, not inside the colonies where bees are present. Or you can also use botanicals or essential oils like neem extract, cinnamon oil, peppermint oil, eucalyptus oil, clove oil, ginger oil, garlic oil. These oils also provide efficient control against mites. Or next pest is ectoparasitic honeybee mites. As I have already mentioned, two types of mites: one Varroa destructor and second Tropalilaps scleri, which is called Asian mite. Uh, they are most common and found almost every beekeeping areas in Pakistan. They in fact they attack on sealed and unsealed brood, which cause death and injury to the bees. They suck the blood or fat bodies from the brood or adult honeybees. Here is the comparison of these two species. The Varroa destructor is 1 mm in length and 1.5 mm wider, whereas the Tropalilaps scleri zero point 0.7 to 1 mm in length and zero point 0.5 to 7 mm in width. Here is the life stages of a Varroa destructor: larva, then larva hatches, then protonymph to deutonymph, and then adult female or adult male. The male are always smaller than the female and lighter in color. There are two stages of the mite. One is the phoretic phase or phoretic stage, and the second is foundress or mother mite. uh, if when a bee emerge, then the matured mites also emerge with her, and the mite attach to the nurse bees and found to search for another cell for the breeding. This phase is called phoretic phase, and it occurs outside the brood cell. When the phoretic mite found a suitable cell for egg laying, uh, and this stage is converted into then foundress phase or mother mite, which is always occur inside the brood cell, and then the mating take place within the cell. Here is the complete life cycle of the Varroa destructor. Before the capping of cell, the matured might enter into the cell and hide under the larva or food. After when the cell capped, they puncture the larvae and start feeding on the larvae. And after the capping, and after 60 to 70 hours of cell capping, the female lay her first egg, which is always of male. And after the egg laying of male egg, after every 30 hours, she lay female eggs. The female might mature in 6 to 7 days, whereas the male mature in 5 to 6 days. And at the day of 21st, when the bee emerged, the matured mated mites also emerged with her, whereas the unmatured mites or males will die when the cell opens. Here is the life stages of Tropalilaps scleri, same as the Varroa destructor: protonymph, deutonymph, and female and male. Here's the life cycle of Tropalilaps scleri. As you can see, at the day of 8 day before the cell capping, the female might enter, and after the cell capping, the first 60 to 70 hours after the cell capping, the first egg she lay a first egg which is of male, and after 30 hours she lay female eggs. And you can see at the day of 21st and the day of 21st, when the bee emerged, all the matured mice also emerge with her. Here are the symptoms which you can identify: either they are attacked by mites or some other pests. the affected mites have deformed wings. It means, as you can see in the picture, the wings are not fully developed. The second symptom is bees. You found bees crawling at the colony entrance, and the most obvious symptom is the spotty brood or perforated capping. As you can see in this second picture, the capping is not properly, the brood is not properly capped. Here in this picture, you can see the attack of varroa mite on different stages of the bees, uh, on larvae, pupae, and adults, and you can see the population of varroa mites within the cell. Here is the damage caused by the Tropaileps scleri. Here you can see on larvae and pupae, adult, and population within the cell. How these mites transfer from one colony to another colony? There are several reasons. One is the honeybee drifting. Why honeybee drift? Maybe if you have placed the colonies near to each other, then it's the chances to honeybee may drift. And the weak colonies are unable to defend their colonies, the strong colonies rob their resources, uh, rob their resources, due to which mites can transfer from one colony, a weak colony to strong colonies. And some bees have their orientation problems, due to which mites can transfer from one colony to another colony, as well as due to the queenlessness. If your box is queenless, then the worker bees might drift to other colonies. And another reason is the robbing. As I have already mentioned, the weak colonies during the when the food resources are limited and flora is not available outside, then the strong colonies attack on the weaker colonies, and weaker colonies are unable to defend. If the weak colonies are affected by the mites, then what happened? The robber bees carry mites from weaker colonies to strong colonies. Moreover, some beekeeping practices due to which mites can transfer from one to another colony, like moving frames. If you transfer the frame from infected colonies to the healthy colonies, the mites can also transfer due to unsanitized equipments like bee brush, hive tool, queen cages, and if you have purchased bee colonies from those apiaries which are already affected by the mites, then you can also transfer mites from the affected apiary to your apiary. And also queen introduction. If you are introducing a new queen, then you can also transfer mites with her. Some other ways are also like swarming. Uh, if if a swarm can occur from a colony which are infested by the mites attack, and you capture the swarm, and after capturing the swarm, you put it in a box and place in your apiary. Then also you can also transfer. Then the mites can transfer from the swarming colony to other colonies. Moreover, mites can also transfer through wind or natural dispersal, and the most important, through floral transmission. The mites attach to the forager bees. When the forager bee landed on a flower to gather nectar and pollen, the mites detach from the bee and wait for an other bee to visit the flower. And when the an other bee visited the flower, they attach that on that bee and transfer to another colony. Before the management of these mites, we should have to monitor the population of mites within the colony. There are three ways to monitor the population of mites. One is the screen bottom board mite tray, and second sugar roll method, and third one is the alcohol wash method. I will show you the pictures of these three methods. If you found if you record 2 mites per day from January to March through screen bottom board mite method, mm, and during April to May, if you record 7 mites per day, and in June to December 8 mites per day, then you should have to adopt the control tactics. And in case of sugar roll and alcohol wash method, if you found 2 to 3 mites per 100 bees in the spring and early summer, and 5 to 6 mites per 100 bees in the late summer and fall, then you have to apply the control measures. Here is the first method, screen bottom board mite tray. Here in the first picture, you can see the screen bottom board mite tray. Put it on the bottom board within the hive, and after 3 to 5 days, remove the screen bottom board from the hive, and count mites from the material. If you found, I have already mentioned, if you found it means 2 mites per day from January to March, and 7 mites from April to May, and 8 mites from June to December, then you have to adopt the control measures. Next is the sugar roll methods. Take 300 nurse bees from the colony, and put in a plastic jar, as you can see in the pictures, and contain the jar containing 200 gram of powdered sugar. Gently roll it for 2 minutes. Gently roll it for 2 minutes, and pour all the material on a white blank page, and then count the mites with the help of lens. The sugar roll method is safer for the bees, as it didn't kill any of the sampled bees, whereas in case of alcohol wash method, it is dangerous method, because it kill all the sampling bees. For the alcohol wash method, you should have alcohol wash tray or some kind of bowl with sieve, and take 300 bees from the nurse bees from the hive, and put 200 ml ethanol, and put 200 ml ethanol, and leave it for 1 to 2 minutes. And as you can see in the picture, pour all the material in a sieve, and you can count mites from the material or ethanol within the tray. How you can control these mites? There are two methods. One is the non-chemical control method, and the second one are chemical control method. In non-chemical control methods, you can use drone brood trapping, in which you provide a drone brood sheet to the colony, as the bees prefer to attack on drone broods, because drone broods have more developmental take more developmental time, because the drone emerged from egg to adult in 24 days, whereas the worker developed in 21 days. So the mites trapped in these drone brood sheets, and but you should have to make sure that remove the drone brood sheet before the opening of the drone brood, or emergence of the drone brood. The second one is the removal of worker attacked worker brood. The third one, queen caging. The queen caging is the biotechnical method for the control of mites. As you can see in the pictures, take a cage, and put the queen in it, and place it within the hive. If you cage the queen for 21 days, it means then during the caging, because queen become unable to lay eggs within the hive, due to which the all the capped or uncapped brood within 21 days emerged, and no due to the absence of the brood within the hive, the mites will die. The fourth method is the sticky bottom board tray, just like screen bottom board tray method. It is the same, but it have a sticky sheets. The safer one of safer method is dusting powder sugar. As you can see in the picture, put powdered sugar on the top of the frames, and with the help of brush put it within the spaces between the between the frames. This is the latest device which is now which is used throughout the world, but not in Pakistan. This device is basically operates on the basis of temperature. If you put the infected brood frames within this chamber and for 2 hours at 40 to 42 degrees Celsius, it will kill all the stages of the mites, either either they are proto-nymph or deutonymph or matured females. But this method is safer for the brood because the larvae can bear up to the temperature of 48 degrees Celsius. Next, chemical control method. There are two types of chemicals which we used for the control of these mites. One is the soft chemicals, which are organic acids like formic acid and oxalic acids, thymol, and essential oils like thymol oil, ginger, garlic, mint, lemongrass, cinnamon, etc. The formic acid is effective against both these species like Bro and Tropalalis. Take 10 ml from the 65% diluted formic acid and put it on a cardboard, absorbent cardboard, and place under on the bottom board of the hive. The formic acid will create the fumes which kill all the mites, either they are on the bees, adult bees, or within the cells. Or you can use 20 to 25 gram thymol in a petri dish. Yeah, or oxalic acid. Oxalic acid, only use oxalic acid in those conditions when the colony is broodless, just like in winter, or you can say dearth period, during dearth period, in either in summer, July or August, when the colony is broodless. The oxalic acid, you can apply oxalic acid by three methods. One is the trickling, second spraying, and third one vaporization. Take 75 gram oxalic acid and dissolve it in 1 kg sugar solution, and apply 10 ml either through trickling method or through direct spraying on the bees. In trickling method, take 10 ml of the solution and put the solution within the spaces between the frames. The third method is the oxalic acid vaporizer. Take 1 gram of oxalic acid and put it into the plunger, and then put the nozzle in hive. It will generate fumes or vapors which will kill all the mites in the colony. Next are the hard chemicals, which are fluvalinate or episton strips, which are most effective for the control of mites. But you have to be ensure that I'll not apply these kinds of chemicals during the honey flow period. Otherwise, their residues will remain in the honey. Recently, we have published the manual for integrated pest management of honeybee mites. It's in our national language Urdu. It is helpful for the beekeepers for the management of mites. Next is the tracheal mite, which is endoparasitic. These mites are microscopic and internal mites of honeybees,
Excuse
which
me, Dr.
attack.
Saab.
Yes.
Sir, if you can wind up your presentation within
Okay.
5
Just
minutes.
give me 5 to 10 minutes.
Yeah, 5 to 10 minutes, please.
Okay.
Thank you.
Okay. Thank you. These mites are capable of infecting all the three individuals within the colony, like workers, drones, and queens. They enter through the prothoracic sphericles of the honeybee and puncture the tracheal wall and feeds on the hemolymph. The infected bees, you found the infected bees crawling in front of the hive, and bees become sluggish, and their flight activity of the forager bees become also reduced. And they also cause Acaropsis disease in honeybees. Here is the complete life cycle. They mostly prefer to attack on young honeybees, which is of 3 to 4 days old. They entered into the bee tracheal system and lay 14 eggs. The mating take place within the tracheal system, and the mated mites emerge emerge uh, em- um, emerge from the emerge from the sphericals and attach to the bee hairs, and due to the close contact among bees can cause female mites from transfer female mites from infected bee to the non-infected bees. For the control of these endoparasitic mites, tracheal mites, you can use 50 gram of crystalline menthol in a plastic bag, which are porous, which will menthol will provide efficient control for the control of these mites. The most dangerous are hornets. They are the most they are the predators of these honeybees. Either they are Apis mellifera or Apis cerana. Yeah, these hornets are almost found almost all the beekeeping areas of Pakistan and cause 20 to 25% of the losses of bee colony. There are 5 types of hornets. The first one is the Vespa orientalis. They make their nests underground. The second one, Vespa tropica, they make their nests in hollow trees or sometime underground. This next one is the Vespa basalis, which is called black-bellied hornets. They make their nest on large trees, and they are the voracious feeder of honeybees. Next is the Vespa velutina. This wasp is found almost all the beekeeping areas of Pakistan, either in AJK and GB, or you can say in Punjab areas, mostly Islamabad and Chakwal area, and you can say KP areas. And a single individual of this hornet can kill 718 bees in just 4 hours. You can imagine how dangerous he is. The next one is the Vespa mandarinia. They also make their nest underground. How you can control? You can use hornet traps for the control of these hornets, as you can see in the picture. Or you can depute some persons for flapping the visitor hornets, or you can use anti-hornet screens at their hive entrance, or always seal the cracks and crevices from the hive so that the hornets are unable to enter into the hive. Or the most safest way is to use this kind of technology. Just locate the nests and burn them. It's the safest way. Next pest is which is minor pest ants. I will not talk about it, because these are the minor pests. So how you can manage these ants? Always seal cracks and crevices, or you can use these ant-proof feeds or ant-proof sticky products, and ant-proof hive stand, or ant-proof hive feed. Next, the birds are you can say in few areas which like in few areas of KP and in Chakwal. These birds are serious pests of honeybees. They prey on honeybees and attack or catch the flying bees. A single green bee eater can kill almost 708 forager honeybees per day. Here are some species of these birds which are found in Pakistan. How you can manage? You can use reflective tapes, or the DVDs which have one side, which have one reflective sides. Just hang the DVDs or these reflective tapes on the trees near your apiary, or you can destroy the nesting sites of the bee-eating birds. Thank you, and I will be happy to answer your questions.
Jazakallah khair, Dr. Wasim. Mashallah, such a knowledgeable and very experience-based presentation that you shared with us today. Now it's
Thank
time
you.
for the question answer session, and I request to our respected participants, if someone want to ask directly to our respected speaker, please raise your hand and ask your question, please. Thank you.
Is there any purity test?
All right, Mr. Amir Hamza, please unmute yourself and ask. Okay, Dr. Wasim, can you check the
Yes.
chat box, please, and see if someone asked a question?
Thank you for your question. Dr. Benjamin, yes, you can ask. Dr. Benjamin have a question, I think. Can you? Naila Sikandar have a question, why it is so difficult to control viral diseases in honeybees? You cannot control the viral disease, but you can stop the spread, but you can stop the spread of the diseases of viral honeybee. Sorry, viral infection, but you can stop. Um, uh, but in Pakistan, I think only 1 to 2 diseases are here in Pakistan. One is the chalk brood, and the second one, you can say Nozema disease. Nozema disease. So, as I have mentioned in my mites lecture, uh, deformed wing virus. As deformed wing virus is only transferred through the mites. So if you control the mites, then you can also control deformed wing virus disease.
Okay. I greet everyone. I appreciate this lecture.
Thank
I've
you.
learned some things today. From my practice in beekeeping in Nigeria, I do encounter the small hive beetle and the wax moth. So my question is, what is the best way I can control the small hive beetle and the wax moth? I don't know if you also have similar experience in Pakistan. So I wish
Uh,
you can share with me. Thank you.
the small hive beetles, there is no attack of small hive beetles in Pakistan. But yes, wax moth is the most dangerous pest in here in Pakistan. But for the control of wax moth, I have already discussed about how you can control the wax moth. So small hive beetle is not here in Pakistan.
Any other question? Dr. Wasim, is there any question in the chat box? Please, you can take from the chat box.
I think no. controlled by disease.
Yeah, Mr. Amir Hamza, would you like to ask a question, please? Either you can
Okay.
send or you can
This is
directly
Amir
ask.
Hamza. Fungal disease.
Assalamu alaikum, Dr. Wasim. First of all, thank you so much for wonderful presentation. My question is basically because my master's research was on fungal disease of honeybees, and I researched on the pathogens I studied were nosema, stone brood, and chalk brood. Although stone brood is very rare, it was only observed during the spring season, and I only observed one or two pathogens. Other than that, they were not present. But chalk brood and nosema, they were quite prevalent, and out of those two, nosema was much more prevalent, and I observed that about 85% of colonies that I experimented in that were infected by nosema. And is that because of the lack of testing facilities and the lack of farmers, like apiculture farmers, interest and their awareness about diseases, or is that is because of the thing that they are quite prevalent in our region?
Yes, there is no control of chalk brood. You can, I think you know, because you are working on it. The chalk brood, you can only control the chalk brood through if you put the colony far from your apiary, or remove all the only we have only you can say non-chemical methods for the control of chalk brood. um, uh, you can say remove you can only remove the chalk brood frames, which are chalk brood frames, and just destroy them. Or you can queen cage the queen for just 21 days. You can also control you can also control the mites as well as chalk brood. So yeah, you are right, most of our beekeepers don't know about it. That's the major reason. Then that's why we here in HBRI and some other organizations conducting training regarding this for the awareness of beekeepers. So I yes, you are right. And for yeah, most of our Apis rana beekeepers are also facing the nosema disease. Uh, and even they cannot distinguish either the sirena are attacked by nosema disease, or which endoparasitic mites, tracheal mites. They cannot identify the attack of which either it is disease or the attack of mites, because the symptoms of nosema disease and tracheal mites are almost similar. But in nosema disease, you can only identify with the help of yellow spots on the frames or outside the hives or on the bottom boards. So yes, um, we should have to arrange the training programs for the awareness of beekeepers. I hope.
Yeah, thank you, Dr. Wasim, for such a knowledgeable presentation and such a fruitful discussion based on your practical experience while working at Honeybee Research Institute, NARC, Islamabad. On behalf of the participants, I just want to ask one to two concluding question. So, as you are the last speaker of this second session of the apiculture entrepreneurship workshop, so firstly, I would like you to say some home tech message or the concluding remarks based on the last 3 or 4 lectures since yesterday. So that not only covering your lecture but also the Khalid Saab, Dr. Sumaira and Miss Amara Baloch And as a concluding home tech message for our respected participants, and what you suggest on behalf, or what you offer on behalf of the Honeybee Research Institute, NARC, Islamabad, Pakistan, not only for the national participants or the students or the stakeholders who are directly or indirectly engage or involved in the apiculture or honeybee farming, but also for the participants joining us globally from different parts of the world, as this honeybee or farming or the apiculture is very high demand business on commercial scale, and everyone who can afford is using the honey, the quality honey in their daily diet. So, what your research institute is offering for the national stakeholders, but as well as for the international stakeholders, and how the HBRI, the NARC, is offering some collaboration or the research opportunities for the national and the international, the one who is interested in this work in research or the trading or goes with the entrepreneurship. Please go for the home tech message, please. Thank you. Dr. Wasim, please unmute yourself, and we want to hear from you.
Sorry, I was I was not here. I was not here. So can you repeat, please?
Okay, you could not hear my. Anyhow, I will
Yes.
repeat briefly, because I took a long time to ask you the this important last question. Or actually, we want, as you are representing the Honeybee Research Institute, NARC, and you are the last speaker, and all the colleagues that presented earlier to you. We want you that on behalf of the HBRI, what you say for the closing remarks for this second session of the apiculture workshop. So we want to hear, or we want to know that what the HBRI is offering for the national stakeholders, including students, researchers, professors, or the industrialists who are directly or indirectly involving in the apiculture sector in Pakistan, but as well how the international participants or those who are interested to collaborate with the Honeybee Research Institute, NARC, Islamabad, at national or international level, how one can approach you, and how what are the possible what are the I just want to say, what are the possible opportunities for the collaboration, not only at national level but also the international level, and what is the home tech message from your side, the HBRI, for this workshop participants, please. Thank you.
Uh, Thank you, Dr. Sakib. First of all, the beekeeping is a livelihood type of business, and I will recommend the our youth to start this business. Because our institute HVRI is the first institute which initiate the business of honeybees in Pakistan. We make the this country as a honey exporting country, and we are the first who introduced Apis Italian bee or Apis mellifera bee in Pakistan in almost 1977. We imported Apis mellifera from you can say Australia, and then we trained our people mostly from KP and AJK to start this to rear or start this kind of business in Pakistan. And we are here for to serve the people of Pakistan either they are from Sindh or Punjab, either they belong from any province or AJK. We are here. We will provide you the you can say any kind of help, or also provide you the permit, especially permit for which is important for during shifting process, shifting of honeybees from one area to another area, as well as we have a collaboration with the Turkish organization. We have international collaborators. Recently, we have here in HBRI, we have introduced a automatic wax foundation sheet plant here, and we are working already. We have working with other universities like NUST, and Dr. Sumera is also working with the Texla University, Engineering University, yeah, and the Islamia University of Bahawalpur. Yes, we are working. Some of the one of my student from Bahauddin Zikria University has now completed his PhD in on integrated pest management of honeybee mites. So we are collaborating internationally and nationally to promote the beekeeping throughout the world or in Pakistan. And we will welcome our foreigners participants to collaborate in this beekeeping business with us. I will inshallah I will provide you the email and my contact number for the further for the future collaboration, inshallah.
Thank you very much, Dr. Wasim, Sir Khalid, Dr. Sumera, and Ms. Amara Baloch from National Agriculture Research Center, Islamabad, Pakistan, that all of you guys spare your valuable time, especially in during weekends, and you speak with us, our respected participants, and share your valuable knowledge related with the apiculture and the entrepreneurship in the in this important sector that actually based on the entomological knowledge. So based on our the objectives of this workshop, our main focus that we actually want to cover every aspect of apiculture through this workshop, because this is the second session. First we did previously maybe 1 or 2 months earlier with us with one speaker who was actually an entrepreneur who started his apiculture business when he was a bachelor student of entomology. Then later on he made this as his as a profession or as a career as a bee apiculture consultant. So keeping in mind the concept that the one who uh, studied the entomology and after bachelor, if he or she can want to start an entomological based startup, so I think the best option is the apiculture, because it's highly demand, a daily consumable commodity, and not only in Pakistan but it has demand globally. So thank you very much, Dr. Wasim, again. And on behalf of the Gen team, as the lead coordinator, I want to say again, thank you very much, our respected speaker, our participants, and we have created a WhatsApp group for as apiculture counseling that are participants joining us globally. And I request you, please, I shared the link. If the one who is really interested in apiculture as science, as entrepreneurship, as business, as trading, want to learn more and more about continuous, if one want to go for the continuous counseling in apiculture, so I request you to please join, not just only to become as a member, but an active participant, that we request you to share the valuable information, knowledge, experience, and the problems you are facing while dealing with the apiary. Either you belong to any city in Pakistan, any town, any location or province, or you are joining us or the member any part of the world. So we want to create a global community of the one who is interested or directly or indirectly involved in apiculture. So we request you to join our group. That's group actually for you, not just only to become a member. So finally, I would like to request the Gen Ambassador, Miss Ansa Ali. Please go for the closing remarks for this second session of the apiculture bootcamp workshop. Thank you, Miss Ansa Ali, please.
On behalf of the Global Entomologists Network, we extend our sincere appreciation to all our distinguished speakers for delivering insightful and inspiring sessions during the workshop titled Bee Business Bootcamp Entrepreneurship in Modern Apiculture. Today's workshop provided valuable scientific and practical insights into modern apiculture entrepreneurship, including sustainable hive management, commercial beekeeping, value-added bee products, quality assurance, and business development strategies. The sessions highlighted how scientific beekeeping practices can be integrated with entrepreneurship to build profitable, sustainable, and environmentally responsible enterprises. The discussion also emphasized emerging opportunities within the apiculture sector and the importance of innovation, market oriented approaches, and professional skills in strengthening bee based businesses and rural livelihoods. We sincerely appreciate the active participation of all attendees, whose engagement and thoughtful discussions contributed greatly to the success of this workshop. At GEN, we remain committed to promoting scientific learning, mentorship, and capacity building among students, researchers, and professionals worldwide. With that, we officially end today's workshop. Stay connected, stay innovative. Thank you all.
Thank you very much and take care. See you next time, inshallah. Assalamu alaikum.